So I'd like to now introduce Carolyn Conant, PhD, who is an application scientist at Fluxion Biosciences, to get started with the webinar.
All right, good day to you. Thank you very much for joining us for our webinar. Today we're going to start off with the agenda. We're going to talk a little bit about the background of Fluxion Biosciences, who we are, followed by a discussion of some of the parameters that are important for dynamic assays that are outlined in the title. We'll then explain the BioFlux system and how it works and all of the components. And finally, we'll look at some of the unique applications of the system to study cellular motility, and then finally what the instrument configuration looks like. Fluxion is a small company based in the San Francisco Bay Area. Our main focus is to design instruments and consumables that enable functional assays for live cells. Currently, we offer products in two spaces. One, for many types of live cell assays using microscopy, and that's the BioFlux. That's what we're going to talk about today. And two, for screening ion channel response to compounds, called BioFlux, which we'd be very happy to answer questions about later or provide more information by request. So if you look on the bottom of the slide, you can see the BioFlux instruments, and then on the right-hand side, you can see the BioFlux instruments. Okay. Our very first system, the BioFlux 200, was launched in 2008 and has been adopted by labs worldwide, both for academic research and for R&D in the pharmaceutical industry. What BioFlux offers and what we're going to talk about today is an added dimension or added dimensions of control over several critical parameters that influence biology and that are not controlled for in traditional live cell or static live cell assays. So some of the controllable variables delivered by the BioFlux are shear, the importance of which we'll discuss in the following slides, maintenance of consistent conditions, for example, compound delivery or environmental conditions like temperature, atmosphere, as well as the ability to perform these in parallel and to collect dynamic data in time-lapse, which is especially important for the assays that we're going to talk about today. The first parameter we're going to discuss is shear. Shear is a mechanical force, in this case, or in our case, exacted by fluid flow. In a healthy individual in the vasculature, shear can range from sub 1 dyne to 20 dynes per centimeter squared. In the case of a stenosed artery or a diseased blood vessel, the shear can be well in excess of 20 to hundreds of dynes per centimeter squared. This ever-present force has a large influence on biological processes, causing changes in gene expression leading to both good changes, like what we might see during embryonic development, and pathological changes leading to things like blood clot formation and inflammation. So it makes sense that shear should be a controlled parameter in many experiments, especially those occurring in the vasculature. Study these areas. Boyden chamber assays do not provide shear. Here is a good example of where shear plays a very important role in vivo. The biology of the endothelium is always under blood flow. When endothelial cells are grown under shear flow, as shown on the right, there are many changes in cell signaling which lead to morphological and functional changes in cell behavior. In some cases, as I said in the previous slide, the response to shear can be beneficial, such as conferring resistance to injury or resistance to vascular permeability. In other cases, shear can lead to very bad things like atherosclerosis and thrombus formation. Besides shear stress, using flow for biological assays confers a number of additional benefits, such as full management of the microenvironment that the cells see, which is important to ensure consistent cell behavior time after time. As well, this provides the opportunity to change conditions reliably to create areas of continuous chemoattractant concentration to enable chemotaxis, for example. And we'll talk about this a little bit in the coming slides. How does the BioFlux do this? It was designed with multi-parametric control in mind. Using the hardware, which we'll talk about shortly, and the microfluidic BioFlux plates, we can tightly control parameters of shear from 0 to 200 dynes per centimeter squared, temperature from ambient to physiological, 37 degrees, even to high physiological, use of external gas hookups, where you might do 5% CO2 to buffer media, or you might go as far as creating anoxic conditions for hypoxia models. And then finally, our unique fluid control interface allows the channels to be compartmentalized so that assays can be run under 2 conditions at once. And that's really how we do these invasion and migration assays. In addition to controllable environmental parameters, the BioFlux was designed to be easy to use out of the box. The consumables are in a well plate format. As such, many assays can be run in parallel, and liquid handling is very easy. Up to 96 assays can be run per controller at the same time. Because we use a microfluidic format, which by nature uses small amounts of reagents, this opens up the possibility of using precious reagents and rare cells to their full potential. At the heart of the BioFlux is the pneumatic controller, which is shown on the upper left. It affords very tight control over fluid handling. And then the controller couples to the microfluidic plate, or we call that a well plate microfluidic device, via an airtight interface, which I'm pointing to right now on the lower part of that panel. Data can then be collected using microscopy, either manual or automated, and we have user-friendly software that enables both seamless data collection with concurrent flow protocols and automated data analysis. So what is a well plate microfluidic, you might ask? Our devices are based on an SBS well plate, which you can see here on the upper left. It serves as a chassis for the microfluidic channels, which are shown on the bottom of the plate here. So this would be the well plate turned over on to have the wells facing down. All the channels run from well to well, and they're all independent units or independent experiments. The channels are then repeated over the entire footprint of the well plate, giving you several experiments per plate. In this case, there's 24 shown. And at the bottom of each plate is a full-size cover glass, which covers the entire bottom, and it's optimized for your inverted microscopy. Here on the lower right-hand side, you can see a cross-sectional view, and this is really the well and the other well, and then you can see the channel flowing underneath. And we'll see this a little bit more in the coming slides. How does a typical experiment work using a BioFlux plate? Again, this is a cross-sectional view of a channel just cut in half down the middle. What we do is dispense coatings and cells into the wells. So you can see some little cartoon cells here. And then the interface is placed on top of the plate, and it makes an airtight seal. Pneumatic pressure is then applied, and fluids or cells in the wells are then pushed into the channel. And then you can do your experiment here, and you can also do your data collection. You can see there's a microscope objective underneath for viewing the experiment. If you look down on the very lower right-hand side of the slide, you can see what the channel looks like if you have a cross-section. So there's 350 microns wide, and then the channel is 75 microns in height. Here are some of the features that make the BioFlux plate or the well plate microfluidic conducive to live cell assays. Again, the well plate makes it easy to handle. You can pipette into it using manual pipetters or automated or even robots. The cover glass, again, is optimized for imaging using inverted microscopy. All the channels are extremely close in size to each other. I consider them identical. And the way the hardware is designed with the interface, the entire experiment is contained within the plate. There's no fluid that's going to leave the plate and go into the tubing, which is traditional in a flow system. Okay, let's just talk quickly about the different devices, and then we'll get on to applications. The plates come in 2 main formats, 24 and 48 well chassis, and each one has a really different purpose. The focus of the webinar today is on the functionality of the 24-well device, which has 8 channels. You can see half of the plate shown here. You can see each channel has 2 inlets and 1 outlet. And if you look down here in the middle where I'm pointing, you can see that the channels, the 2 inlets meet inside this area called the viewing window, and you can see that you have 4 channels ganged together. And this plate works in physiological shear range. So just quickly, I'll show the 48-well plate, but just by way of introduction, and then we'll go back to the 24-well plate. The 48-well plate comes in 2 different shear ranges, enabling both physiological shear, or lower flow rates, and pathological shear, or higher flow rates. Each channel, unlike the 24-well plate, is formed of only 2 wells, so an inlet well and an outlet well. And this enables a higher throughput per plate and more data points. So let's move on. Critical to understanding the assays we're going to discuss today are the characteristics of the microfluidic channels. First is the concept of laminar flow, which you may or may not be familiar with. So if you look at this, this is a channel from the bird's eye view, and you can see there are some arrows there. And that's to indicate that the channels have laminar flow. In fluid dynamics, what this means is that the flow is extremely orderly within the device, and that if you have particles, they're traveling through the channel as if there are cars in lanes on a highway, meaning, well, in the case where the cars aren't passing each other, but the flow is very orderly and things are not crossing streamlines. This enables precise handling of conditions in the channel. In addition, if you look on the right side of the slide, both the flow velocity and the shear within the experimental area are designed to be uniform, and these are just some in silico models of what both the velocity on the top and the shear at the bottom wall look like. Okay, back to the 24-well device. Flow can either be controlled from intermittent inlet wells, meaning flow from one inlet or the other, in which case you might want to switch from a buffer to a compound or something like that, or 2 different nutrient conditions in the same experiment. Or the flow can be from parallel wells, meaning that the 2 inlets are running at the same time together. Due to the fluid dynamic conditions that I explained in the previous slide, within the channels, the 2 fluids remain compartmentalized in discrete sides of the channel. And that's what you see down here on the lower right. This is actually an experiment where we flowed green fluorescent beads from one inlet and red fluorescent beads from the other inlet in parallel. And what you can see is that there are two discrete compartments formed by using this type of flow, and that there is no mixing between the red and green beads. So again, we call this parallel flow. In the case of assays in the BioFlux system, we can use the parallel flow to make 2 fluid compartments, or we can partition the channel into a liquid and a solid zone, and we'll talk about that for invasion assays. Okay, so the next slide has a video, which will take a few minutes to load, but I'll give you a preview of it as it's loading. First, what you're going to see are the wells being loaded with a red compound red, and a blue compound. And you'll see this from a cross-sectional view. In the next part of the video, what you'll see is it's as if you were looking in a microscope, so a bird's eye or a bottom view. And what you'll observe in the video is that when you flow from the 2 inlets, you can flow the fluids one at a time, as I said, intermittent flow. So in this case, it will go the blue compound and the red compound. Or you can flow in parallel, so you'll see the red and the blue flowing together. And parallel flow is what we use for chemotaxis, migration, and invasion assays. So it looks like a fair number of attendees have the video loaded, so I will begin to play it. So here you can see the compound loaded in the well. This is the way we operate the plate. And now you can see the microscope view of the blue compound, the red compound. And now you'll see what I mean by parallel flow, so the 2 flows together. Okay. We've done a lot of work to characterize this type of fluidic compartmentalization in the channels. In this case, we've done an experiment where we have loaded a fluorescent dye in one of the inlets and a non-fluorescent buffer into the other inlet, both of which have the same viscosity. So what we did then is vary the pressure on the inlet containing the non-fluorescent buffer, which is inlet B in this case. So in this graph, what you can see are the resulting highly reproducible conditions over 14 different channels. And these are measurements of the compartment size that contains the fluorescent dye. What you can see is that the size of the compartment is identical from channel to channel. This is actually a mean flow of 14 different channels. And above the graph, in each one of the micrographs, you can see the size of the compartment that we can get by varying the shear. So we have very tight control over the conditions, and we can very reproducibly make these fluid compartments, which is very conducive to doing all sorts of assays. So let's now switch gears and talk about some of the applications. And these are all going to be focused on cell motility and migration. Many, many types of cells are screened for their motility in response to compounds in chambers called Boyden chambers. You may also know them as transwell inserts. So what you can see is a little cross-sectional diagram. And the idea behind this type of assay is that you're creating two compartments. So the bottom compartment may contain a chemokine, it may contain a conditioned media, and the top compartment contains cells. And usually there's a porous membrane in between. In the case of invasion assays, there'd actually be a deposition of gel here. And the idea is to understand how the cells are responding in the two compartments. So these are really providing a static data point in an otherwise dynamic process. You can only understand what's happening at the end point of an experiment. And what we do in the BioFlux is using the parallel flow capability, we can actually follow the entire process that you look at in a Boyden chamber, and we can screen that in the channel for applications, as I said before, migration invasion and endothelial cell invasion. We can really look at every single step of the process in real time. Shown here, just to summarize, are some special cases of cell motility in response to stimuli. All the phenomena are important in health and disease, and each step of each process can be targeted for drug development. Therefore, gaining insight into all of the temporal events comprising these phenomena is really crucial to understanding both the basic science and also developing treatments for any of the diseases that are involved in these phenomena. First, let's start with chemotaxis. The phenomenon of chemotaxis can be thought of as the response to a call to action by a cell. In this example, this cartoon shown here, this is a normal response to an injury in the tissue. And what's happening is because of the injury, there are chemoattractants being secreted into the vasculature, and the neutrophil is being attracted to the site of injury to go clean up the mess. And all of these steps we can actually study in the BioFlux. The one that we're going to focus on today is the actual movement of the cell from one place to another. Okay. Studying chemotaxis in the BioFlux channels involves flowing a chemokine in through one side of the channel, and usually, in most cases, another fluid from the other inlet, typically buffer or growth media from the other side of the channel. Data are captured over time during the entire time of the experiment. In this case, we're talking about an hour. And the movement of the cells in response to the stimulus can be tracked. So in this particular example, we're studying the effect of FMLP, which is a very potent chemokine, on cells which are neutrophilic differentiated HL60 cells. And these cells are known to respond to FMLP. So the video on the left is a case where we have a channel flowing buffer in the top side and buffer in the bottom side. So we shouldn't expect any type of cell movement other than random. So I'll begin to play the video. So hopefully you can see that cells are moving quite a lot and flow is from left to right, but there's actually no directed movement of the cells other than with the flow. Now, in the case of the video on the right, what we now have is the FMLP being perfused from one inlet, which is shown on the top, and buffer is being perfused from the lower inlet. And what we should see in this video is that the cells are moving both with the flow but also towards the FMLP on the top of the video. So you can actually follow cells moving all the way from the bottom to the top. I'm going to play it once more. So you can look at some of these guys, this one in particular moving up. Oh, I think I lost the pointer. Okay, so both of these videos are 45-minute time lapses. So we've actually measured the data from several of these experiments in this slide. And this is, again, the chemotaxis assay. We're flowing FMLP in one side and buffer in the other. And we're actually doing a dose response. So we're flowing several orders of magnitude of concentration of FMLP in each one of the channels. So we collected time-lapse data of this response over an hour in time-lapse. And actually, you can see here in the upper left-hand corner, this is a micrograph at the beginning of one of the time lapses. And what I've done is overlay the trajectories of the cells. In this case, the FMLP is on the bottom compartment. And you can see the cells are starting, they're in their starting point, but if you follow the little lines, that's where they've moved. They all move towards the FMLP. And we're able to follow these data and get a a dose response of the FMLP response. We can also follow other parameters such as angular trajectory and movement and velocity. One example is shown on the lower left-hand side. These black boxes are movement plots or directional plots of these experiments. So let me explain this a little bit. In the center of each crosshair is a white box. That indicates the origin of a particular cell in the experiment. And the direction of movement, in this case on the very left-hand side, is buffer on the top and buffer on the bottom, and flow is from left to right again. And each one of these colored tracks is the actual XY coordinate of where a cell is moved. And in this case, they've moved randomly because there's no stimulus. Then if you look at the black box next to it to the right, you can see again the origin is set. And in this case, there's a chemokine in the top and buffer in the bottom, and flow is from left to right. And you can see that all of the cells are orienting and moving in the direction of the chemokine, as well as somewhat with the flow. So if we look at the dose response here on the upper right, what you can see is we're going from 0 nanomolar FMLP all the way to 10,000 nanomolar. And the orange-colored bars are the percentage of cells that are in the different compartments. So the dark orange is in the buffer compartment, and the more pumpkin-colored orange is in the chemokine compartment, and you can see there's a dose response. And again, I've plotted this on the bottom just as a line plot, and you can see there's a dose response here. Okay, so let's switch gears and start to talk about invasion assays. For invasion assays, we can use parallel flow to create a gel region inside the channel as if you were using a Boyden chamber but turned on its side. The gel can be dosed with compounds or growth factors, as well as the fluid can be dosed with compounds or growth factors. And then again, we can look at the cellular response followed in time lapse. So just to show you, here on the left-hand side, there is a channel with gel patterned. You can see the gel, a very light stripe in the middle where the gel is contained from the middle to the top of the field of view. And then on the lower part, you can see the buffer. And on the micrograph on the right, you can see I actually put beads into the gel. So now the gel's on the lower part, and you can see the beads. And then I'm flowing fluorescent beads in the fluid part, and you can see the beads are streaking by in the flow, and the beads on the bottom are not moving. The first cell invasion example I'm going to talk about is angiogenesis. Angiogenesis is a multi-step process that starts out relatively simply, really with a single cell. So you can see this cell on the left here. What happens is the cell, just like in chemotaxis, it gets a chemical message and then it responds to that message by starting the process of angiogenesis, which is a very complex process we'll look at in the next slide. And eventually, this single cell can actually lead to a newly formed blood vessel. And I actually have included on the right-hand side the worst case scenario of angiogenesis, which is a tumor that's been highly vascularized. This is one strategy for tumors to really continue their life, is to attract lots of blood vessels, and they can feed themselves and grow bigger and bigger. As you can imagine, just like many processes that occur in biology, this is a very highly complex process and consists of many discrete steps, beginning, of course, with the cellular invasion of the cell out of the blood vessel into the surrounding tissue. So if you look at some of these steps, we start with release of proteases. We have to destroy the basal lamina. The cells then migrate, there's cell proliferation, and then you look at the steps of the blood vessel lumen formation, and then eventually you have initiation of blood flow. If you look at the steps that are highlighted in blue, these are the ones that we can look at in the BioFlux. So degradation of the matrix and movement of the endothelial cells into the interstitial area. The BioFlux has a very important advantage for studying these early steps of angiogenesis, aside from looking at in the Boyden chamber. We have the ability to add time-lapse, use physiological shear, and add and subtract growth factors as we need to. Shear is very important to this process, and it's been shown in the literature using very similar methods with collagen plugs that when you're using shear, you actually get better invasion into the matrix than you would in static conditions or with just growth factors. So what's shown on the right-hand side, these micrographs are actually the endothelial cells sprouting into the collagen gel. You can see that they've moved quite a lot further than the cells on the far right, and this is a paper from Kang et al. And then I just included a cartoon here on the left, which is showing some of the different responses during angiogenesis. So you can see on the top there's some sprouting, and then below there's some branching. And all of these we can observe in the BioFlux system. Interestingly enough, many of the steps of angiogenesis, and why I really pointed them out, is many of them are actually target areas for therapeutics. And in the case, again, the first three of them can be studied in the BioFlux system. Okay. In the BioFlux, what we've done here is we've used Matrigel in one half of the channel, and then we flow media in the other half. And in this case, what we did was we introduced some primary endothelial cells into the fluid compartment of the channel, and we were able to follow the cell's response to the matrix under the cell's response as they've attached to the matrix and then invade the matrix and so on and so forth. In case of this experiment, within about 2 hours, almost all of the endothelial cells had degraded and invaded into the matrix. And in the case shown here on the lower right-hand side, we actually put fumagillin into the gel when we patterned it into the channels. And this is an angiogenesis inhibitor. And what we found is that the cells could only line up along the gel and they couldn't invade into it. If you move up from the bottom to the top on the right, what you can see is the gel is on the top stained green, and you can see an endothelial cell completely inside the gel making some sprouts. In the case of the one in the middle, we've put FGF in, and the cell is also making sprouts and is invaded in. This is actually at the end point of the experiment, and the cells have undergone apoptosis because of the media conditions. But you can really look at every step of this in time lapse and observe both sprouting, process formation, and apoptosis. Another example where you can study cell invasion is with cancer cells. So metastasis involves the movement of cancer cells from the primary tumor to other sites in the body. Again, this is a dynamic, multi-step process. Not every step has been completely researched and studied, and there are a lot of unknowns in this area. And we can look at a lot of these steps in the BioFlux system. Shown here are the time-lapse data for two types of cancer cell lines. One is HT1080, which is a colon carcinoma cell line, and the other is MCF7, which is a commonly used breast cancer cell line. So what these are time-lapse montages of, I believe each one is an hour, and the channel is shown, and so I'm going to try to point to where the gel is. It's patterned in the bottom half of the channel, and the cells have been introduced into the top half. So if you're able to follow this through the time, what you can see is around 4 hours, the cells begin to invade the matrix, and you can then continue to follow this over time. By the end of this time lapse, which is 15 hours in total, you can see that the cells have started to invade and they're beginning to form structures in the gel. If you look at the cells on the right-hand side, which are the MCF7 cells, these are non-invasive cells. So we've, of course, put the cells into the channel. The gel is, again, on the bottom part of the channel. And if you follow through the time lapse for this one, what you can see is that the cells are never invading the gel, and this is a great control for this experiment. The next slide has a video, so I'll just give you a preview of it as it's loading. This is the actual time lapse that I showed the montage from the previous slide. So this will be the HT1080 cells, and they have already been perfused into one side of the channel. We have some low shear flow to exchange the media in this case, and then the bottom part of the channel again is patterned with Matrigel. And so this time lapse goes for about 15 hours, and you'll see the cells begin to invade as well as divide in the gel. So it's almost fully loaded. I'll just give it one more second. And I will now play it. So what you can see is the cells are lining up along the gel fluid interface, and then they actually will begin to very vigorously move into the gel and even begin to divide within 15 hours. Shown here is the actual quantitation of the time-lapse data for these cells. This is a measurement of cell invasiveness over time, where the dark orange line is showing the amount of gel that's been occupied by HT1080 cells, and the scale on the x-axis is time. And then the light orange, which is lining up along the x-axis, is actually the data for the MCF7 cells, which basically have 0% invasion, and they cannot invade the matrix. The advantages of using the BioFlux system are that data from several conditions can be captured at once in time-lapse, so you could do many migration or chemotaxis assays in parallel. You can use one system to study all of these different things, both the fluid-fluid setup or the fluid-solid setup where we did invasion. Data capture is only limited to the limits of what your microscope can do, so you don't have to do any kind of handling. You're really doing everything inside the channel. Everything is in situ, which in comparison to a Boyden chamber eliminates the need to fix cells, handle the membranes, cut the membranes off the transwells, mount slides, all of those things. So you can really observe this live and even without labeling them. Finally, all of these phenomena occur in the vasculature in the presence of shear. So if you need the shear is delivered by the BioFlux. To further summarize the experimental parameters that we discussed today for migration and invasion assays, number one, we leverage the parallel flow capabilities of the BioFlux to create unique two-compartment experiments that can be observed in one dimension. And two, These can be performed with 2 liquid or a liquid flow stream abutting a solid matrix. So that concludes our discussion of applications. Here are the 2 product offerings for BioFlux. On the top, you'll see the BioFlux 200 system, which consists of the controller, software, and accessories, everything you need to run your assays if you already have your own microscope. And on the bottom, you can see the BioFlux 1000, which is a fully automated and integrated system that includes automated image capture and all the hardware and software that's needed to run and walk-away automation. If you have further interest in the system, would like more information about the system or applications, please visit fluxionbio.com, or to inquire about ordering, please contact our sales department at sales@fluxionbio.com or at 866-266-8380. Thank you very much